Welcome. This is the oral presentation for a study on UV and hydrogen peroxide treatment for chloramphenicol degradation, specifically focusing on the process analysis and optimization. This study was conducted by Michele Giulietti, Antonio Zuorro, Roberto Lavecchia, Sergio Garcia Segura, and Monica Brienza. To introduce the main topic of this study, in the last decades, antibiotic contamination in the environment has become a major engineering challenge due to their large-scale production, widespread use, and incorrect disposal. The antibiotic of concern for this study is chloramphenicol, which has been used as antibiotic in preventative treatment for food-producing animals and domestic poultry. The structure of the antibiotic can be seen in the figure to the right. Less than 70% of chloramphenicol can be metabolized after its intake by humans or animals, which results in a high amount of the antibiotic released to aquatic environments through urine or feces. Chloramphenicol is also a recalcitrant compound and cannot be removed by traditional wastewater treatment processes, which calls for concern. Due to various inefficiencies of physical and biological processes for the treatment of chloramphenicol, promising applied technologies for chemical degradation include advanced oxidation processes, or AOPs. Advanced oxidation processes are based on the generation of radical species, such as hydroxide, at ambient temperature and pressure. These methods seem to be the most promising because they allow the complete mineralization of many contaminants in simple inorganic compounds such as water and carbon dioxide. A previous study has evaluated the photon-driven degradation of chloramphenicol with the combined use of hydrogen peroxide and UVC radiation. This, in aqueous effluents, induces photolysis, which yields hydroxyl radicals that in turn destroy compounds into their transformation products. In addition to being used for the abatement of organic compounds, advanced oxidation processes are also used to decrease the level of toxicity and to study new products that can be generated through these processes. For the experimental conditions, all experiments were run in a photochemical cabinet, which was maintained at 20 degrees Celsius, which was equipped with a UVC irradiation system, as can be seen on the bottom left figure. For the kinetic study portion, the concentration of chloramphenicol was monitored by spectrophotometer absorbance at 290 nanometers of wavelength in order to avoid interference with the hydrogen peroxide. This can be seen on the top right figure. The transformation of products, however, were studied with the use of high-precision liquid chromatography, or HPLC, as can be seen on the bottom right figure. In the main method used, cells with a volume of 4 milliliter were filled with an antibiotic solution at the desired concentration and hydrogen peroxide concentrations. And these cuvettes were placed in the reactor at the desired distance to control a UV radiation dose. The quartz cuvette used can be seen in the top left figure, while the setup again can be viewed in the bottom left figure. The samples were collected and immediately analyzed at the beginning of the experiment, and then after 10, 30, 60, and 90 minutes of exposure to radiation, in order to observe the effects of the process. The first results that we obtained were the preliminary trials, and they were performed prior to the experiment in order to determine the effect of direct oxidation by hydrogen peroxide without UV radiation and the effect of photolysis by UV radiation without hydrogen peroxide. The chloramphenicol degradation always started at a concentration of 50 milligrams per liter in our experiments. The percentage of degradation of the antibiotic, which was used in the study, is defined by the equation on the top right, where the difference between the initial and final concentration of antibiotic is divided by the initial concentration of antibiotic in the cells and made into a percentage. The results were that the direct oxidation processes of only hydrogen peroxide and no UV radiation showed no significant effect on the degradation of the antibiotic after 40 minutes. However, the effect of photolysis by UV radiation without hydrogen peroxide showed very little effect, less than 1% removal, in fact. However, trials with the combined use of UV radiation at 1,000 microwatts per centimeter squared and 40 millimoles of hydrogen peroxide achieved 99% degradation of the 50 milligrams per liter of chloramphenicol within 40 minutes. And this can be seen in the figure on the right. The second portion of the results regarded the modeling and optimization. Different polynomial models were tested for their ability to fit the experimental data. An essential composite design was used to evaluate the effects of initial chloramphenicol concentration, hydrogen peroxide levels, UV intensity, reaction time, and pH on the degradation of the antibiotic. The best result was obtained with the full quadratic model, which was refined by a stepwise procedure to include only the statistically significant predictors. The optimal degradation condition parameters that were found at the end of the experiments can be seen on the bottom left of the slide. For a chloramphenicol concentration of 50 milligrams per liters, a combined treatment of 25 millimoles of hydrogen peroxide, 1,240 microwatts per centimeter squared of UV radiation for a time of 60 minutes at pH of 6.2. And this would yield chloramphenicol conversion of about 97.2%. In the figure on the right, we represented some sample results from trials which were evaluating the effect of different hydrogen peroxide concentrations on the degradation of the antibiotic. And this can be observed in the figure on the right. Lastly, the last portion of the results regarded the pathway degradation, and it was hypothesized by the identification of carboxylic acid in the solution and then 4 unknown main transformation byproducts. The stable aromatic byproducts that were formed during the UVC and hydrogen peroxide treatment were identified by reverse phase high-precision liquid chromatography analysis of aliquots collected at different photon-driven treatment times. The byproducts identified included oxalic, malic, maleic, and formic acid, and the associated results can be seen in the figure on the right, where all 4 byproducts are graphed. As can be seen by the bottom portion of the graphs, malic acid, which is the blue curve, was the first low molecular weight carboxylic acid byproduct released as a result of the chloramphenicol degradation. In conclusion, the main findings of this study are that the photodegradation process can be represented by first-order kinetics, with rate constant values and quantum yields that varied depending on the pH of the solution. The statistical model that was developed provided useful indications on the contribution of the most important process parameters, alone or in combination, on the degradation of chloramphenicol, as well as on the optimum set of treatment conditions needed. Additionally, both chloramphenicol and the main products were totally degraded in less than 60 minutes, but the mineralization based on the total organic compound analysis was only 30% due to the persistence of carboxylic acids. Thank you very much for your time.